Hello friends, welcome to SAM online class. Friends, today we shall discuss about the structure and function of plasma membrane. Both prokaryotic cell and eukaryotic cell are enclosed by ultra-thin membrane known as plasma membrane or cell membrane or plasma lemma. The plasma membrane is ultra-thin, fragile. It is a protein lipid sheet. The main component of the plasma membrane are protein and lipid. In addition to protein and lipid, a small amount of carbohydrate are also present in the plasma membrane, and these carbohydrates are associated with protein and lipid. In the plasma membrane, there is a lipid bilayer. The lipid bilayer it acts as the structural backbone of the plasma membrane. The lipid bilayer it acts as a barrier. It prevents the random movement of water-soluble molecules into and out of the cell. Chemical composition of plasma membrane. The main components present in a plasma membrane are lipid, protein and carbohydrate. The ratio of lipid to protein in the plasma membrane varies depending on the type of organism and also on the type of cell. The plasma membrane contains a wide diversity of lipids. The lipids that are present in the cell membrane can be divided into three main classes. One is phosphoglyceride, second is sphingolipid, third is sterol. All such lipids that are present in the cell membrane are amphipathic. Amphipathic means such lipids contain both polar region and non-polar region. First we shall discuss about phosphoglycerides. Phosphoglycerides are a type of phospholipid that contain glycerol backbone. Two hydroxyl groups of the glycerol are esterified with two molecules of fatty acid, and the third hydroxyl group is esterified with a phosphate group. The phosphate group in turn is associated with additional groups. The additional groups can be choline, ethanolamine, serine or inositol. So, various phosphoglycerides that are present in the plasma membrane are phosphatidyl-choline, phosphatidyl-ethanolamine, phosphatidyl-serine and phosphatidyl-inositol. The phosphate group along with the additional groups that are associated with this, they form a highly polar head group. Second type of lipid that is present in the plasma membrane is sphingolipids. Sphingolipids are less abundant as compared to phosphoglycerides, and all sphingolipids are the derivatives of sphingosine. Sphingosine is an amino alcohol having a long hydrocarbon chain. In sphingolipids, the amino group of sphingosine is associated with a fatty acid, and the hydroxyl group that is present in the sphingosine is associated with additional group. If the additional group is phosphorylcholine, then that type of sphingolipid is called as sphingomyelin. In some other sphingolipids, carbohydrates are also associated with the hydroxyl group of sphingosine. If carbohydrates are the additional groups, then such type of lipids are called as glycolipids. Glycolipids are usually present in the outer leaflet of plasma membrane. So, in many glycolipids, the associated carbohydrate consists of simple sugar. Such type of glycolipids are called as cerebroside. In some glycolipid, the carbohydrate moiety consists of a small cluster of sugars. Such type of glycolipids are called as gangliosides. Third type of lipid that is present in cell membrane is sterol. Sterol is a steroid alcohol. Different type of sterols are found in the plasma membrane of different organisms. For example, in animal cell, cholesterol is present in the cell membrane, whereas in plants and other organisms, different type of sterols are present. So these three different type of lipids together, they constitute the lipid bilayer. And the lipid bilayer, it is the structural backbone of the cell membrane. Second most important component is proteins. Proteins that are associated with the lipid bilayer, they perform specific functions. Some of the proteins are also associated with carbohydrates. Such type of proteins are called as glycoproteins. The proteins that are present in the cell membrane can be divided into three different groups. One is integral or transmembrane protein. Second is peripheral membrane protein. Third is lipid anchored protein. First, integral or transmembrane protein. Integral or transmembrane protein, they are embedded in the lipid bilayer, and such proteins cross from one side of the membrane to the other side. Second type of protein is peripheral protein. Peripheral proteins are present on the outside of the lipid bilayer. Peripheral proteins may be present on the cytoplasmic side or extracellular side. And these peripheral membrane proteins are associated with the membrane surface by non-covalent bonds. Another type of protein is known as lipid anchored proteins. Lipid anchored proteins are a specific group of proteins that are associated with the membrane lipids with the help of covalent bond. So the third component is carbohydrate. A small amount of carbohydrate is also present in the cell membrane, and the amount of carbohydrate accounts for 2 to 10% by weight. And the carbohydrates that are present in the cell membrane face toward the extracellular space. And out of the total amount of carbohydrate, 80% of the carbohydrate is associated with protein, and remaining 20% of carbohydrate is associated with lipid. And the carbohydrate that is associated with protein is usually oligosaccharides. Such oligosaccharides contain less than 15 sugars per chain. And in such glycoproteins, the carbohydrate moiety is linked with the protein part with the help of two types of glycosidic bond. One is N-glycosidic linkage and the other is O-glycosidic linkage. So friends, this is about the chemical composition of cell membrane. Several models have been proposed to explain the structure of cell membrane. Out of them, the widely accepted model is fluid mosaic model. The fluid mosaic model was proposed by Sinder and Nicholson in 1972. According to fluid mosaic model, the cell membrane is a quasi-fluid structure. That means the cell membrane has fluidity. The fluidity and the elasticity of the cell membrane is due to the presence of a lipid bilayer. This lipid bilayer is the structural backbone of the cell membrane and also other biological membrane. So the cell membrane is a two-dimensional fluid. In such a two-dimensional fluid, lipid and other molecules can diffuse laterally. Even these molecules can also spin within the lipid bilayer. So according to fluid mosaic model, the cell membrane consists of a lipid bilayer in which protein molecules are embedded. The lipid bilayer means two layers of lipid together constitute the lipid bilayer. Most of the lipids that are present in the cell membrane are amphipathic. That means they have both hydrophilic part and non-hydrophilic part. The hydrophilic part remain exposed to the external surface or to the internal surface. So these circles represent the hydrophilic head group or polar head group. The lipid molecules are arranged in the form of a bilayer in such a manner that the hydrophilic head group remain oriented toward the external side or internal side. So these are the polar head groups and this is the hydrophobic tail. In this lipid bilayer, the interior is hydrophobic, and in this lipid bilayer you can also see the presence of sterol. For example, in animal cell membrane, cholesterol is present, and some of the lipid molecules are also associated with carbohydrate. These are the oligosaccharide chains, and such type of lipids which are associated with carbohydrate are called as glycolipids. And in this lipid bilayer, protein molecules are also embedded. These proteins are divided into two different groups on the basis of their association with the lipid bilayer. They are peripheral membrane protein and integral membrane protein. Peripheral membrane protein remain associated with the surface of the lipid bilayer. These are the peripheral membrane protein. They are composed of a large number of hydrophilic amino acids. The second type of membrane protein is integral membrane proteins. The peripheral membrane proteins, because they are loosely associated with the surface, so they can be easily removed. Whereas integral membrane protein are associated permanently with the lipid bilayer. Some of the integral membrane protein do not cross the lipid bilayer. That means they remain embedded only in part of the lipid bilayer. So this is a type of integral membrane protein which do not span the membrane. And other type of integral membrane protein span the entirety of the cell membrane. These are the integral membrane protein which span the lipid bilayer. Such type of integral membrane proteins are called as transmembrane proteins. The protein components that are associated with the lipid bilayer in the cell membrane perform special function, for example, transport. The transmembrane protein, they can span the lipid bilayer once or more than once. If the transmembrane protein span the lipid bilayer once, that type of protein is known as single span protein. Whereas there are many transmembrane protein span the lipid bilayer several times. Such type of integral membrane protein are called as multi-span protein. So the transmembrane protein consists of a middle hydrophobic region and two outer hydrophilic regions. The hydrophilic regions that remain on the outer side, it interact with the external side or internal side, whereas the hydrophobic region remain embedded in the lipid bilayer. And the transmembrane protein that are present in the cell membrane can be divided into two different types. One is alpha helical protein and the other is beta valence. So in addition to lipid and protein, there are also small amount of carbohydrate that remain associated with the lipid to form glycolipid, and carbohydrate may also be associated with protein to form glycoprotein. So in the cell membrane there is a lipid bilayer. All the lipids are amphipathic. The polar head group remain exposed to the outer side or inner side, whereas the hydrophobic tail remain within the interior of the lipid bilayer. And in this lipid bilayer, you can also see the presence of different type of proteins. Two type of proteins are there in the cell membrane: peripheral membrane protein and integral membrane protein. Also, small amount of carbohydrate remain associated in the form of oligosaccharide chain, either with the lipid or with the protein. So friends, this is the structure of cell membrane. Functions of cell membrane. First function is protection. The cell membrane physically separates the intracellular components from the extracellular environment. By preventing the entry and exit of specific substances, it helps to maintain a constant internal environment. Second function is maintain cell shape. The cell membrane helps in anchoring the cytoskeleton, thereby providing shape to the cell. Third function is selective permeability. The cell membrane is selectively permeable. That means it allows specific substances to pass into the cell and out of the cell. The selective permeability of the cell membrane is due to the presence of different type of transport proteins which help in the transport of molecules and ions. Fourth function is cell potential. The cell membrane also helps to maintain a cell potential, and the cell potential is necessary to carry out various functions in specific type of cell. Fifth function is transport. The cell membrane can also transport a variety of substances, charged and uncharged, from one side to other side, either along the concentration gradient or against the electrochemical gradient. Two types of transport can occur across the cell membrane. They are passive transport and active transport. Passive transport takes place along the concentration gradient, so there is no involvement of metabolic energy. Passive transport can occur either by simple diffusion or by facilitated diffusion. Other form of transport is active transport. Active transport leads to the accumulation of molecules or ions inside the cell. Active transport takes place against an electrochemical gradient, so metabolic energy is needed. The metabolic energy comes from the hydrolysis of ATP, or the energy can also comes from an ion gradient or from some other source. So in active transport, molecules and ions are transported against the gradient from one side to the other side of the cell. And the active transport is of two types: primary active transport and secondary active transport. In primary active transport, energy primarily comes from the hydrolysis of ATP, whereas in secondary active transport, the energy is derived from an already existing ion gradient. Next function is Endocytosis. Endocytosis is a process in which various cells internalize solid substances, small molecules and ions, and even macromolecules by engulfing them. The engulfment of solid substances is known as phagocytosis or cell eating. Similarly, cells can also internalize small molecules and ions. That process is known as pinocytosis or cell drinking. The process of endocytosis is an active process, so it needs input of energy to drive the transport. Other function is exocytosis. Exocytosis is a process by which cells can remove undigested and toxic substances from the cell to the outside. By this process, the cells can also secrete specific substances. So, in the process of exocytosis, the membrane of a vesicle fuses with the cell membrane. After that, the content that are present inside the vesicle are released to the outside. This process is known as exocytosis. So, with the help of exocytosis, a cell is able to remove undigested substances of the components that are brought in by the process of endocytosis. It is also helpful in secretion of specific substances to the outer environment. Next function is cell signaling. It is also known as cell communication. Cell signaling is the ability of a cell to receive, transmit and to process various signals with its environment or with itself. The signals that a cell receives from outside can be a physical agent or a chemical agent, and specific proteins that are present on the cell membrane act as a receptor for such type of signal. Last function is cell-cell recognition. The living cells have the capacity to distinguish between different types of cells. This ability is due to the presence of specific molecules on the surface of various cells. When the receptors that are present on a cell bind with the ligands that are present on the surface of other cells, then interaction occurs between the receptor and the ligand. This interaction between the receptor and ligand, it initiates a cascade of events that causes a change in the behavior of the cell. So, through cell-to-cell recognition, a living organism is able to distinguish between self and non-self. So friends, this is about the chemical composition, structure and function of plasma membrane. For more update, please subscribe my YouTube channel, Sam Online Class. Thank you very much.